Today we will discuss about the structure of tRNA. tRNA means transfer RNA because it helps in the transfer of amino acids in protein biosynthesis. So the name is tRNA, transfer RNA. It is also known as soluble RNA because it dissolves in 1 molar NaCl. It is also known as soluble RNA because it dissolves in 1 molar NaCl. And it is the smallest RNA. tRNA is the smallest RNA and it is single-stranded RNA. As the other RNA, tRNA is also single-stranded. And about 15% of the total RNA is tRNA. 15% of the total RNA is tRNA. And the main function of tRNA is it acts as a specific carriers of activated amino acids in protein biosynthesis. It acts as a specific carriers of activated amino acids in protein biosynthesis. Now coming to the common structural features of tRNA. Common structural features of tRNA. All tRNA molecules have a common design and consist of 3 folds. All tRNA molecules have a common design and consist of 3 folds, giving it a shape of clover leaf. Giving it a shape of the clover leaf with 4 arms. A clover leaf shape with 4 arms. These are the arms. 1, 2, 3, 4. And in the case of longer tRNAs, there will be a short 5th or extra arm. This is the extra arm. There will be a short 5th or extra arm in the longer tRNAs. The actual 3-dimensional structure of tRNA looks like a twisted L. The actual 3-dimensional structure looks like a twisted L. Twisted L. tRNA molecules are unbranched. tRNA molecules are unbranched and consist of 73 to 93 ribonucleotides. 73 to 93 ribonucleotides. They are unbranched. And the molecular weight is in between 24,000 and 31,000. The molecular weight is in between 24,000 and 31,000. tRNA contain 7 to 15 unusual modified bases. tRNA contain 7 to 15 unusual modified bases. They are methylated or dimethylated adenine, uracil, guanine and cytosine. Methylated or dimethylated adenine, uracil, guanine and cytosine. Pseudouridine and 5-methyl cytosine are the common examples for the unusual bases. Pseudouridine and 5-methyl cytosine are the common example for the unusual bases. In this, pseudouridine is the most abundant unusual base. Pseudouridine is the most abundant unusual base in the tRNA. The 5' end is phosphorylated. The 5' end, this is the 5' end it is phosphorylated. Usually the guanylate residue is there. At the 5' end, usually guanylate residue is present and that will be phosphorylated. The 5' end is phosphorylated. And the 3' end is CCA. The 3' end is CCA and to the 3' of the ribose, the 3' of the ribose, the amino acid is binding in the protein biosynthesis. The base sequence at the 3' end is CCA and the 3' of this CCA binds the amino acids for the protein biosynthesis. So this is known as amino acid attachment site or amino acid binding site. The 3' OH is known as amino acid binding site. the 3' and CCA sequence is present. CCA base sequences are present always in the 3' end of the tRNA, and this act as the amino acid binding site. And the binding site is the 3' OH group of the ribose. 3' OH group of the ribose. So the linear structure of tRNA is like this. At the 5' end there is a phosphorylated guanine and the base sequences are there, and at the 3' end CCA is present at the 3' end. This is the linear structure of tRNA. 50% of the bases are paired. They are like this. 50% of the bases are paired and forming double helix like this. But 5 groups of bases are not base paired. 5 groups of bases not in base paired. They are 3' CCA terminal. This 3' CCA terminal and T psi C loop. This one T psi C loop, and the extra arm. This is the extra arm, and DHU loop, dihydrouridine loop. This one, and then anticodon loop. This. These loops are not base paired. The loops act as recognition sites. The loops act as the recognition sites. So this is the 5' end of the tRNA, and this is the 3' end of tRNA. The 5' end is phosphorylated. Always there will be a guanine residue, guanylated residue. So the 5' end is phosphorylated, and the 3' end, this is the 3' end. It contains CCA bases. The base sequences are CCA. Here the 3' OH of the last adenine is the binding site for the amino acid. The 3' OH of the adenine is the binding site for the amino acid. So this is known as amino acid binding site or amino acid attachment site. So the transfer of the amino acid is carried out by binding the amino acid here, and it transfers the amino acid to the protein biosynthesis. The lowermost arm or loop is known as anticodon loop. The lowermost arm or loop is known as anticodon loop because it contains anticodes. It is known as anticodon loop because it contains anticodes. Consider an mRNA which contains code AUG. This tRNA has to read this code AUG and attach the corresponding amino acid to the 3' OH of the CCA, 3' OH at the amino acid binding site, and bring it to the mRNA. So the anticodon will be here UAC. UAC. The corresponding anticodon to this AUG is UAC. So due to the presence of this anticodon, this loop is known as anticodon loop. Anticodon loop. This loop is known as T psi C loop. T psi C loop or T loop. T psi C loop or T loop. This psi means pseudouridine or pseudouracil. The psi means pseudouridine or pseudouracil. Pseudouridine or pseudouracil. So because of the presence of this psi, this and presence of the thymine, Usually the thymine is not present in the RNA, but here thymine is present. So this arm is known as tRNA arm or tRNA loop or T loop. And the function of this loop is ribosome recognition. Ribosome recognition. So this is known as ribosome recognition site. Ribosome recognition site. Now this is known as D loop or dihydrouridine loop, because the unusual base dihydrouridine or uracil is present here. And the function of this site is amino acid recognition. Amino acid recognition. So this is the amino acid recognition site. Amino acid recognition site, also known as DHU loop or D loop. Now let us summarize the structure of tRNA. This is the 5' end and this is the 3' end. The 5' end always contain a phosphorylated guanine. Phosphorylated guanine. And the 3' OH contain CCA base sequence. CCA base sequence, and to the 3' OH of the adenine is the binding site of amino acid. So this is known as the amino acid binding site. Amino acid binding site. There are 4 arms in the tRNA and 3 loops are there. 3 loops are there. One is tRNA loop, one is anticodon loop, and one is dihydrouridine loop. In the tRNA loop, thymine, pseudouridine and cytosine is present. Thymine, pseudouridine and cytosine is present. So the name is tRNA loop or T loop. And this is the ribosome recognition site. tRNA loop is the ribosome recognition site. And next is anticodon loop, which contain the anticodon corresponding to the codons of the mRNA. Corresponding to the codons of the mRNA. So this name is anticodon loop. And this is D loop, also known as dihydrouridine loop, because of the presence of the dihydrouridine or dihydrouracil. Dihydrouridine or dihydrouracil. And it is the amino acid recognition site. It is the amino acid recognition site. So this anticodon loop read the codons in the mRNA. Read the codons in the mRNA, and this DHU loop recognizes the amino acid, and that amino acid is binds to this amino acid binding site 3' OH of the CCA, and the tRNA loop recognizes the ribosome where it has to bind. So all these things work in coordination during the protein synthesis and transfer the amino acid corresponding to the mRNA to the ribosome. This is about the structure of tRNA. Thank you.